So good afternoon, everyone, and thank you to Gail and Georgina for inviting me to do this talk. I think I've given a talk to the PPIA panel maybe about a couple of years ago. So it's nice to come back and tell you a bit more about some of the work that's progressed over the last 18 months and the impact it's having nationally. Some of this is work in progress. So I'm literally sharing with you data prior to publication. So a lot of this is going to be pre-publication, but also how it's changing what NHS England is thinking about in terms of doing clinical trials. So I'll first start with sort of just giving you a bit of background about the work that I do. So this is a summary of the talk today. I'm going to tell you about the information that's buried in what we call a whole cancer genome, and that is sequencing the entire 3,000 million base pairs of the human genome, of the human cancer genome. I'm going to highlight something called mutational signatures, which is a subject matter of mine that's been our focus for 15 years now, and how we use machine learning and AI methods on mutational signatures to create algorithms that help us interpret the data. And then I'm going to end on telling you about its unpublished work where we're integrating that genomic data with mortality statistics that have been collected nationally to understand how genomics data can be used more effectively, and then how that is changing our perception of how to perform clinical trials going forward. Okay, so there's quite a lot there, but I thought I'll just start here, and I do apologize if I know this is Cambridge. Many people already probably know a lot about this, but I thought I would start here because we all started life as a single cell, a single fertilized egg, a combination of genetic information from our moms and dads. But that single copy of a human genome has to multiply many thousands of times to form a baby that will grow up to adulthood, and then who will become an adult sitting here listening to me on this call. And every time your cells divide, your cells are acquiring mutations. Now, if you look at a cancer genome, the DNA of a cancer, and you compare it to the original genome that you were conceived with, the cancer has now acquired tens of thousands of genetic changes that we call mutations. And this talk is going to be about all those mutations. So just to give you a visual on it, our human genome is packaged into 23 pairs of chromosomes. And here you've got chromosomes 1, 2, 3, 4, 5 in the colored pairs. All the way down to here, we've got 2 X chromosomes. A boy will have an X and a Y. If I were to sequence a cancer genome, this is what I'm going to see. And just at this very crude scale, you can see that there's 3 copies of chromosome 1. It's got a bit of pink on it. It's got a bit of somebody else's chromosome on it. There's 7 copies of chromosome 7. So there's a lot of abnormalities at this very crude scale. If I look at it at a very high resolution level, I'm going to find even more mutations. So this is an example of a cartoon of a chromosome. I'm now going to unravel this chromosome all the way down to the individual DNA building blocks. And of these DNA building blocks, we can have mutations of all sorts. So the mutations that you're seeing in terms of chromosomes, you call those chromosome copy number changes, but there's many different types of mutations. Substitutions are when you change one DNA building block for another. Insertions and deletions does what it says on the tin. You might have some bits that are removed or some that are added. And rearrangements are when you actually break certain parts of the genome and it relocates elsewhere. So these are all just different ways of damaging the genome and producing a change, a mutation. When you do whole genome sequencing, you can see all of it. We don't just have to look in the genes, and we don't have to just look in specific regions. We can look at the whole genome and see all the mutations that are present in each person's cancer. Now, for the last 4 decades, we have focused on mutations in genes because genes are supposed to be the important bits. They're supposed to be the bits that make the proteins. But there's 20,000 genes, but it only makes up 2% of the whole genome. 98% of the whole genome is what we call sort of dark matter, and people often think it's not very important. And so for 4 decades, our focus has just been on genes. And that's because mutations in genes are believed sometimes to be driver mutations. They're believed to be the ones that cause cancer. And so for 4 decades, we have focused on finding driver mutations so that they can become targets for therapeutic intervention. We design drugs against those mutations. And here are some examples of mutations that have been developed through research into these drugs that you can see here on the right-hand side that today we use in clinic in the UK. So here are some of these examples. However, beyond those driver mutations, in the 98% of the dark matter of the genome are many other thousands of mutations, thousands and thousands, right? Because it's 98% of the genome. And when I started my PhD, everybody called them passenger mutations, meaning they were sort of just there for as passengers. They were useless. They were not very informative, and people called it noise. But like anything in biology, things very rarely happen very randomly. And in fact, you can study these passenger mutations and understand them better. So if you study, this is me sort of saying, making the point that, if you actually look at mutational noise, if you look at, things in the bin, you can actually learn quite a lot about how people live. So, if you rummage around somebody's bin, you might find out they're eating too many pizzas or drinking too much wine. So you can actually learn quite a lot by going through the noise, through what we have called mutational noise. So here's our fertilized egg again, and I'm going to argue that as we went from a fertilized egg into developing a cancer, and this is a sort of a dummy drawing of a cancer tree, that while we acquired those mutations were just errors that happened because as our cells divide, they will just acquire certain errors, and that would happen in a very particular way and produce a very particular pattern. But you could be somebody who smokes or uses a tanning bed, and that might actually damage DNA a very particular way, and that might produce its own pattern of mutations. Now, you might have an autoimmune disease or some kind of abnormality that produces bursts of mutations, and that might produce a particular pattern of mutations. Or there might be things that we don't fully understand, but they produce patterns of mutations. So each one of these things we call a mutational process. It will produce a characteristic imprint on the genome, a characteristic pattern that we call a mutational signature. But when you look at all the mutations together, yes, it looks like a random mess of mutational noise. But in fact, if you study many tumors together, you might be able to see these individual patterns. You might even be able to quantify those patterns, and they might be able to tell you something about why someone developed cancer. So, for example, if I look at a tumor look at a lung tumor, I might actually be able to see the tobacco-associated patterns in that lung cancer that tells me that tobacco was a contributor to that cancer. Likewise, I might be able to see the patterns associated with UV damage from a skin cancer as well. And so these mutational signatures, these mutational patterns has become the subject of our research, and and this is just to show you some examples of those mutation patterns. There's about 150 patterns today, but I'm just showing you some examples. These 3 examples, signatures 1, 2, and 3, were the first 3 we ever found. Signature 1 is the signature that's happening as cells divide all the time. So if I could sequence your cells right now, in fact, as you sit here listening to me speak, you are acquiring signature 1. Signature 1 happens because DNA is sitting in oxygen and water. Oxygen and water are the most mutagenic elements in the world, but our cells need them to survive. And so it's a bit of an irony that DNA sits in oxygen and water. It will just mutate, whatever the case. So signature 1 is a fabulous example of just what happens to our cells as we sit here in oxygen and water. Signatures 2 and 13 are signatures that arise because of the activity of an enzyme called ApoBex. ApoBex actually evolved to fight off viruses. But every time they see a bit of DNA that shouldn't be there, they munch it up, and when you can see that in human cancers as well. So this is just to show you different patterns. They look different to each other, and those are the patterns that we try to derive from cancers. And here are 3 other patterns. I hope you can see they look very different to these previous ones, which are from within the cells. These three are from outside the body. So the top one is UV light. Signature 4 is tobacco smoke. We see that in lung cancers. And signature 22 is aristolochic acid, and we see that in liver and kidney cancers. So today, when I sequence a cancer genome in the NHS, I look for these signatures and I can tell when someone has had exposure to some of these things. So aristolochic acid, the one at the bottom, is a fantastic example of a compound that is banned in the West, but it is present in the sort of surrounding, in the environment, in the Balkan states, and it is present in traditional Chinese medicines in the East. And so if I look at tumors from Taiwan and China, I can see liver and kidney cancers full of signature 22. I don't see that in the UK. In fact, in the UK, I've only seen 3 patients with the signature 22. So you can actually tell when someone's had exposure to something that they really shouldn't have had exposure to. Okay, so I refer to these mutational signatures. It is a very abstract concept, and sometimes people call it DNA graffiti, because something has come along and made a mark all over the genome. And actually, DNA graffiti can be very recognizable. So this is a guy who's very famous for producing these Marvel Comics DNA graffiti across New York. This is a guy who produces, again, he produces DNA graffiti around in and around Europe. And his style is very distinctive. So even though nobody sees him doing it, we can all tell that it's his work. And it's the same thing with mutational signatures. And this is a very famous bit of DNA graffiti that happened in London, if I'm not mistaken. These happen on sort of shop shutters. And in fact, I think this was a gift that David Cameron presented with Barack Obama. And actually, there's a message, and this is using that same artist, Ben Ein. There's actually a message in that bit of DNA graffiti. And then, of course, everybody knows Banksy, the top right, again, top left, sorry. Yeah, a very recognizable DNA graffiti. So we know who's been there. This is like a whodunit. So the mutational signatures are used exactly in this way. It's almost a whodunit, what's happened in this person's cancer. Okay, so I hope I've communicated these two things, that there's a lot of information buried in whole cancer genomes. And this idea of mutational signatures, it's a principle, it's a concept, an idea that can only come from the fact that we've got whole genome sequencing data, that I can look at the entire genomic sequence and work with the thousands of mutations. It's big data analysis across patients. I now just want to show you some whole genome data from real patients. So these are 3 different patients with breast cancer. And what you're looking at is all the whole genome data that's present in each of these women. They're 3 unrelated women. And the one in the middle, she is the kind of typical patient that does well with surgery and sort of minor endocrine therapy. The one on the right-hand side does well with surgery, but she requires a specific therapy against HER2. That's trastuzumab. The one on the left is an ER-negative tumor. These patients tend to do a little bit less well than the ER-positive tumors. And this tumor, if you look at it, it looks very different. It's got a very particular abnormality that we call a BRCA1 deficiency. Now, that woman on the left has inherited her BRCA1 mutation. She carries a genetic defect that makes her more likely to develop breast and ovarian cancer. And that's called a mutation in BRCA1. Now, I'm showing you now 2 additional women in the middle on the right. They also have some kind of BRCA1 abnormality, but how they have developed the BRCA1 abnormality is different. So the patient in the middle hasn't inherited a BRCA1 mutation. She has acquired her BRCA1 mutation at some point during teenage years or early adulthood. And the one on the right-hand side does not have any genetic defect, and she has acquired something called an epigenetic defect. The genetics are all fine, but there's some other mechanism that turns off her BRCA1 gene. Now, if I told you that these women do not share a single mutation in common, and that's one of the things that when I started doing this, a lot of people said whole genome sequencing will never work, Serena, it's too complicated. Everybody's genome is so individual and so different. You'll never make any sense out of it. But I'm hoping that when I'm showing you this, you can see that these 3 women have overall whole genomes that have a lot of similarity. They have a lot of the green lines in the middle. They have some of the pink stuff around the green lines. And then the sort of the dots on the outside are largely black, a bit of red, and a bit of gray in it. These are BRCA1 defective cancers that have overall these holistic genome profiles that are similar. And it doesn't matter how they have acquired the abnormality of BRCA1 and the fact that every single mutation is different. Collectively, the pattern, the overall image, the overall holistic profile is very similar to each other. And this is a principle that we use for machine learning and AI. If you give AI and machine learning these sorts of big data and you say, try to find me features that are similar or that are contrasting between these two groups and help me make an algorithm that I can use for future patients, this is exactly what it needs. So this is just to sort of say the mutational signatures are shared across the BRCA1 and 2 patients. And we absolutely contrasted the BRCA1 patients on the right-hand side with tumors on the left, which are ER-positive patients that do well. We took this through a machine learning process, and we developed an algorithm called HR Detect to help us identify future patients that might have BRCA1 and BRCA2 deficiencies. So you'll be saying, well, why have you done that? And the reason we've done that is that BRCA1 and BRCA2 patients, they tend to be sensitive to a drug called PARP inhibitors and to other things like platinum. But our oncologists tell us that it feels like there are many more patients who might have BRCA1 and 2 deficiencies, but they haven't necessarily inherited it. So the first drugs like PARP inhibitors were created for the people who had inherited BRCA1 and 2 defects. But as I showed you earlier, there are also women who don't have inherited BRCA1 and 2 defects. They acquire it, or they turn off BRCA1 and 2 in different ways. And so we believe that those patients will also be sensitive to the same drugs. So that's why we create this algorithm. So I'm giving you this example. We've done this for lots of different things in cancer genomes, but this is one of those examples. And what we found was when we take our algorithm and we apply it across, and this was the first set of whole breast cancer genomes in the 2016, 2017, to our surprise, anything above 0.7 is considered a high score. That's quite a lot of patients that are above that high score. So on the horizontal, we have 560 patients, and on the vertical, we have their scores. And what you can see is many patients breach the blue line, and it's actually 22% of the cohort here. So that's 1 in 5 breast cancers, which is a lot. And indeed, when we look at that group, we find that some of them have indeed got inherited BRCA1 and 2 mutations, but many of them have acquired them, or they have the patterns and we don't know why they have the patterns. So that was 2017. We now know why some of them do have those patterns, but the point is that there really are many more patients that could have things like BRCA1, 2 defects that we can find through our algorithms, through our machine learning algorithms, if we do whole genome sequencing. So this is the sort of the new data, right? We looked at breast cancer genomes from across England. These were patients recruited from all of these sites across England, so there was no site that was left out. And they all were whole genome sequenced in a central facility, and they were all analyzed in the same way. There were just over 3,000 to start off with, and then in the end, we finished the analyses, but also because every patient has an NHS number, we can link it up to hospital episode statistics, what happens in the hospital, what drugs they have, or what treatments they have, what kinds of tumors they have. In addition to that, we can link it up to what has happened to them in terms of mortality. So we get that from the Office of National Statistics. All of this is done anonymously. I just see the whole genome data and I get the hospital episode statistics and mortality information. I don't know who the patients are. I just have all that information entirely anonymously. And that's all I need. I don't need to know who the patients are. I then have this incredible data. There's no other data set that's like this. I have this data, and if I do find anything and I want to check that it's real and it can be validated in another cohort, I have got a fantastic set of collaborators in Sweden called the ScanB project. They have several hundred patients. They don't have thousands, but it's still very useful to check our findings. There are also many data sets that have been published already. They may not be as well curated and annotated as ScanB or as this cohort in the UK. And by the way, this UK cohort is the Genomics England cohort of the 100,000 Genomes project. So anyway, this is just to communicate to you that this is a massive effort. Not I get to talk about it, but actually a lot of people were involved in collecting the samples from all over England. And I have also got them matched to hospital statistics and mortality statistics, and it's all anonymous. Okay, so I'm just gonna tell you sort of the punchline because I want to make sure we have enough time for questions. So I'm going to show you that we have over 2,500 patients, and ER negative and ER positive, which is the kinds of breast cancers, which mirrors what happens in the population. So in general, most women have ER positive tumors, and about 15% have ER negative. So that's what you're seeing on that slide. And I'm only showing you some important information. I can't show you all the data because it's too much of it. But we have clinical information, what kind of grade, what kind of stage. This just tells us something about the aggressiveness of the cancer. Some important drivers, the mutations in genes that I mentioned right at the start. And then all the signatures that I talked about, those patterns, so DNA graffiti. So I've got substitution patterns, I've got indel patterns, I've got structural rearrangement patterns, and then I can add on all my other algorithms if I really wanted to. And the main message I wanted to get across, and actually there's many, but the one I want to get across is HR deficiency, that BRCA1, BRCA2 thing I mentioned earlier. This is a population representative cohort. And what we're finding is that 12%, about 12% are HR deficient. That's still a lot of patients. At the moment, we are, BRCA1 to inherited mutations are believed to be 1 to 2% of breast cancers, not 12%. And that means that there can be really quite a lot of patients out there who don't get the treatments that they probably could and should be getting if we knew and if we have done whole genomes on them. So just to reinforce, we did the study on this left-hand side on England, patients collected between 2013 to 2018. And remember, we have the Swedish study on the right-hand side, and they've got, again, population coverage of ER positive breast cancers. Now, Sweden is a long, narrow country. The black and red dots are where the hospitals are. Some reviewers of our work said, well, why are the hospitals only in the south of Sweden? And the reason the hospitals are only in the south of Sweden is because all the people are in the south of Sweden. Only bears are in the north part of Sweden. So most of the hospitals are down south. But basically, we have two population studies. We have seen what we have seen in England. We went to Sweden and we said, what do we see in Sweden? And in Sweden, we see exactly the same thing. About 12 to 13% of the breast cancer population have got HR deficiency too. So what I want to communicate is this. If I were to do a whole genome sequence in the UK population today, and this pie chart on the left-hand side here, let's say this is the UK population. Today, if I do that whole genome, the slice that has the yellow outline, those patients could have a clinically important mutation or mutation pattern or signature for which there is a targetable treatment today, or that they can go into a clinical trial today to find out whether a specific therapy could work for them. Currently, we don't see these patients and we don't treat them to the level of granularity that I can see in this pie chart today. That's 27% of all breast cancers, which translates to 15,000 patients per year. In the purple slice are the signatures, are the patterns that I know from experimental work are things that could be sensitive to particular drugs. And we are doing preclinical work, so work either in mice or in cells to understand them, but they're ready for going into early clinical trials in patients. So we're going to call the purple slice translational research potential, and that is another 15% of all breast cancers in England, which is 8,500 patients per year. So the bottom line is, if I do a whole genome sequence today on all breast cancers coming into clinic, there's, a lot of patients, 42% of the cohort could either be given a targetable therapy, which they're not potentially getting at the moment, or go into a clinical trial that is phase 3 or even an earlier phase 1-2 trial already today. And so this was the data that has changed our view of whole genome sequencing. Because until this point, in general, when we do clinical trials, we don't do a whole genome. We just look for one mutation. We ask, is someone mutated in this gene or not? We don't look at the rest of it. We just look at one mutation in thousands. And so I'm going to come on to why we need to change that mental concept. So I hope I've covered the first two lines earlier. Now I've covered the machine learning algorithms and how we integrate whole genome data with mortality statistics to get those new insights, and now how we want to change clinical trials. Because every single patient's tumor looks very, very different, and there is so much information that's buried in them that we're currently just ignoring. We're not using this information, which to me is mad. It's like going out for a drive, trying to go somewhere new and not using the latest GPS maps, right? You're not using Waze or Google Maps. You're just going to use some landmarks which are very, very old. And I'm going to show you two patients where the whole genome mattered. And I have permission from the patients to tell their stories. So this is a patient, a young man, a 30-year-old man with a tumor called an angiosarcoma. It's a really rare tumor of the blood vessel lining. And actually, I'm sorry, there's so much information on here. I really want you to just take away the little blue and pink bars on the top right-hand corner, and the big red arrows are pointing at them. This patient's tumor had a signature, had a pattern that is associated with something called polymerase epsilon mutations. And others, not myself, others have shown that polymerase epsilon mutated cancers are sensitive to a drug, an immunotherapy called pembrolizumab. Now, this is a long time ago, 2018. Pembrolizumab was not widely used in this country, so we battled to get the drug for him. So this is him. He in 2017 was found to have this angiosarcoma. It was on his eyebrow, and we removed it. He's got a skin flap there over his eyebrow. But you can see that over the next 18 months, he deteriorated. He didn't respond at all to his angiosarcoma therapy. By July 2018, this man is dying. So he's got mets everywhere. He's got tumors in his lymph nodes, in his chest, in his liver, in his abdomen, in his skull. I mean, he had it everywhere, and he couldn't leave the hospital. And we fought to get him the pembrolizumab because whole genome sequencing hadn't been used before for this. So anyway, he got the pembrolizumab. So these are his scans. The sort of the big gray bits are tumor. And within 3 cycles, everything is melting away. Now, bear in mind, this patient could not walk in July 2018 because his lungs would deflate every time he got out of bed. And by the end of 7 cycles, everything had melted away and he walked home, and he's still alive today. So, this is a nice example of how using a whole genome and looking and reading the whole thing, not just looking at one mutation, reading the whole genome, we actually influence the treatment of this patient. And this is my second case study. This is a Cambridge patient. He's 55 years old, and he had this weird tumor in his thigh, which was thought to be a pleomorphic sarcoma. And he just didn't respond to any of the treatments. He had disease progression. And when we looked at his whole genome, we figured out that he had an abnormality, a bit like HR deficiency, like BRCA1/2 deficiency. This is called mismatch repair deficiency. And these tumors are also sensitive to immunotherapies. It was a battle, but we did manage to get him the treatments that he needed. And again, this man was so sick, he could barely walk downstairs, never mind upstairs. And this is a picture of him attending his niece's wedding, where he was able to go dancing and enjoy the day. I will say this, that my oncology colleagues find it difficult to use whole genome data to treat patients because they do not have the clinical evidence and the guidelines to support them. So the science is all great, but if we want to get it to patients, we need to traverse that translational piece. We need to do the clinical studies to help our oncologists so that they can say, they've done that study, we can use a whole genome, and I can give the patient this drug. That's the piece that we really need to do next. And so what we're trying to push for today is to modernize how we do clinical trials. The UK is famous for actually being one of the failing countries in the world in terms of doing clinical trials. And currently, England is now divided into these 7 domains, 7 genomic medicine services. And we do some sequencing, not everybody gets offered it. Some sequencing analysis happens centrally in Genomics England, where the data sits, and then it pushes back to the genomic medicine services. But as I said, the oncologists cannot act on the whole genomes because they do not feel that there is clinical evidence to support the use of whole genomes yet today. And what we want to do is make those trials happen so that they have that clinical evidence base. So what we'd like to do is permit the genomic medicine services to recruit patients into clinical trials. We need to do the clinical trials, and we need to lean on the current networks that are available in the UK. So Cancer Research UK has a fantastic experimental cancer medicine network. And we'd like to be able to do the trials linked up to the genomic sequencing that happens through whole genomes, because whole genomes are being done in the NHS, we're just not acting on the results. So I'm just going to finish off with showing you this. Today, this is what happens in clinical trials. These are 3 different trials, top, the middle, and the bottom. They're independent trials. They're done independent of each other. You have to have some kind of selection criteria. And then you have the different trials happening. The data is siloed. Nobody shares the data, and there's no learning from each other at all. It's expensive. It takes time, and it takes people. It takes patience. And you put patients through all these individual trials. It's kind of a loss of time, and it's a loss of human energy, really. And what we'd like to do is this instead, that you have a selection criteria and a selection based on the whole genome upfront. So you do the whole genome in the NHS, you get a whole genome, and using that whole genome profile, you slot patients into multiple trials that are going in parallel. So instead of each trial trying to do its own little test on one mutation, we do a whole genome and we go, here's all the information. These patients are HR deficient. These patients are mismatch repair deficient. These patients have HER2 positivity, slot them into all the individual trials. That is the new way we'd like to do this strategy. The stakeholders in discussion right now include NHS England's Director of Cancer, Peter Johnson, NHS England's Director of Genomics Unit, Sue Hill, the Genomic Medicine Services, Genomics England, CRUK, the British Breast Group, the UK Clinical Study Group, many notable academics across the country. This has been a huge exercise trying to get everybody to the table and to agree to work in this way, and critically also partnering with pharmaceutical companies who will help to provide the drugs to allow this to happen. I tell you what we don't have. We don't have a patient voice at the table at the minute. And I think that is the one thing that will make this absolutely happen. If patients say, you know what, we need to do clinical trials in a different way, then, I think this is absolutely going to happen. So this is where we are. Thank you so much for sort of listening to me rabbit on about what has become sort of a passionate thing that actually now many people are involved in our efforts to try to change the landscape and to try to do clinical trials a little bit more effectively. So with that, I'm going to acknowledge the patients, the families, and all the people that have contributed samples. I get to tell the stories, but lots of other people have done. Thousands of people have done this work from across the country. I have some very fantastic collaborators from all over the world. My team are a phenomenal bunch that come from all over the world as well. This picture of ours was actually from 2 years ago out in Hobson's Park, just outside the hospital there. And the ones highlighted in red are the ones who did a huge amount of the work. So I'm going to stop there. Thank you very much. And I'm happy to take any questions. Thank you.